Hello and welcome to Nikolay's genetics lessons, and today's problem is you have to determine the amino acid sequence of the peptide. You perform the following steps using enzyme cleavage of your peptide. Step 1. Treatment with trypsin yields 3 fragments with the following sequences in order of their length: 1, 2, 3 sequences, and each letter here stands for one amino acid. And step 2. Treatment with chymotrypsin gave the following 3 peptide fragments and 1, 2, 3 fragments of the different sequence. What is the sequence of your peptide? According to our problem, our first treatment with trypsin provided us with these 3 fragments of the peptide. So here's one fragment, here's another one, and here's a third fragment. And we can arrange these 3 fragments into following order. For example, let's say that this empty box would stand for the first fragment of the sequence, this is for the second, and this is for the third. In order to find how many different variants we might get, for example, how many variants we can fill the first box. It can be this sequence, this, or this. So we have 3 variants. Let's say we took this sequence in the middle. So in order to fill the second empty box, now we have these 2 sequences to choose from. And let's say we choose this one. So we had 2 variants to choose from. And in order to fill the third box, we have only 1 variant left and no any other choice, so 1 variant here. Now when we multiply all these combinations, we are going to get 6. So we can arrange these 3 fragments into 6 different combinations. How we are going to find the sequence of this polypeptide? And at the step 2, we got different variants of the fragments. Now we are going to use this second step in order to find what fragments of the first treatment and second treatment would have overlaps. When we did second treatment, we got following fragments. So let's take a look what overlaps of this sequence we have with this sequence. And right away I see that we have here 1 overlap, and now we can put this sequence here, and we can put this sequence here, and we can put this sequence here, but we shouldn't get any overhangs in our sequence. So let's move this fragment here, it also fits here, and let's move this fragment here. Now we got the correct sequence of this polypeptide. And this is all for today. Subscribe and see you in the next video. Goodbye.